Hello. Today we are going to go over the essential steps in neurotransmission, as well as one example of how transmission can be inhibited by tetrodotoxin. Before we go into the details of how neurotransmission is processed, we'll first label the key components of the neuron cell. Here we have a diagram of a neuron. In the upper portion we have extending dendrites that act as the receiver for impulses from other neurons. Then we have the nucleus and the cell body that mainly directs the function of the neuron. As the electrical signal is received, the signal is transmitted through the axon and sent to nearby neurons through the synaptic terminal. The mentioned process will now be described in detail. Specifically in the axon, there are voltage-gated sodium and potassium ion channels that line the membrane. At resting potential, the sodium and potassium channels are initially closed to maintain a negative potential on the inside of the neuron. As a neuron impulse is received, a sodium channel opens, allowing a flow of sodium ions to enter the axon. The opening of the first channel stimulates the opening of a chain of sodium channels. As the next sodium channels open to let sodium from the extracellular fluid enter, potassium ion channels open to counteract the influx of positive ions. When the potassium channels open, it causes the sodium ion channels to close, stopping the increase in membrane potential and decreasing it through the potassium ion channels. The neuron transfers potassium at a rate that exceeds the resting potential of the neuron. This can be called the refractory period or the undershoot during the process of neurotransmission. Eventually, the potassium channels close and the membrane potential is restored to its resting potential. The alternating opening and closing of potassium and sodium channels are used to carry the electrical signal a neuron receives to other neurons nearby.
As you can see from the membrane potential graph shown, a threshold potential, indicated by the dashed line, must be attained in order for the action potential to be fully executed. At first, the electrical signal is received, causing an influx of sodium into the neuron, denoted by the numbers 2 and 3. Once the action potential is reached, the signal is sent through the neuron network, and the system then loses its membrane potential through the potassium ion channels, undershooting but later returning back to the resting potential. This cycle repeats until the signal arrives at its target.
Next, we'll help you visualize what happens after the signal is sent through the axon, specifically the intricate interactions that occur at the synaptic terminal. When the electrical signal is sent down the axon, an influx of calcium triggered by this signal causes vesicles containing neurotransmitters to move toward the presynaptic membrane with which the vesicles will fuse. This causes the release of neurotransmitters to the synaptic cleft, where they will bind to ligand-gated ion channels located on the postsynaptic membrane, effectively transferring electrical signals from one neuron to the next. There are many different types of neurotransmitters, the most common being glutamate, which is present in over 90% of all brain synapses. It's a vital neurotransmitter for learning and memory. There are numerous ways in which the nervous system can be damaged. For example, tetrodotoxin, a potent neurotoxin that can be found in pufferfish, acts as an inhibitor to sodium ion channels. In the presence of tetrodotoxin, sodium channels cannot open, thus prohibiting the rising phase of the membrane potential. This can lead to devastating consequences, such as severe numbness and paralysis of muscles in the body. I hope you guys learned something about neurotransmission. Thank you all for watching.